Okay, guys. Good evening. I will talk a little bit about chapter 1, and I did a lot of notes here, but it's 25 minutes, so we cannot go deeper. But I will explain all the things all the chapter 1 is about, and some specific topics. Okay. First we have in this book, chapter 1, themes in physiology, and says that physiology is an integrative science. So let's check what is this, because we have levels of organization, organ systems integration, and some themes, specific themes. Levels of organization is about, for example, in this part, it's about chemistry, molecular biology, cell biology, physiology, and ecology. And all of this together can generate a lot of data and can influence each other and help each other with data, acquiring by experiments or studies or something else. Okay. As they say, the integration of function across many levels of organization is a special focus on physiology. Also, it means to integrate everything to work together and to generate data. It's about two organ systems and the human body and their integration. For example, here we have a schematic figure showing some systems of the body, the big ones. And the interesting thing is that if something occurs in one of the systems and they are interrogated, it can affect other systems in the body. So, for example, body fluid volume influences blood pressure, while changes in blood pressure alter kidney function because the kidney regulates fluid. It means that if something occurs in your blood pressure, there's consequence in other parts of your body. So everything is interligated to work together and for better function. Okay. And we have some function and mechanisms. There's a slight difference between function and mechanisms because function is the why something happened. So why blood carry oxygen? Because every tissues and every organs needs the oxygen to work. So this is the why. This is the function of the blood. Okay, and the mechanism is how it works. The oxygen binds to the red blood cells, and then the blood will carry the oxygen to all the tissues and organs and everything that works with blood. Okay. So it's divided into structure and function are closely related because we have the function, but the structure of the cell or something like this is dependent of their function. Molecular interactions it means that the exchange between, for example, extra cells fluid and intracells fluid. And the compartmentation is about membranes that the spaces into the cell, and each part has a function. So the structure defines the function, or function defines the structure. Living organisms need energy. Every organism needs to grow. So the growth, reproduction, movement, homeostasis, everything needs energy. This energy is on demand. So if an organism needs to make reactions, they will need energy from food, or like in the plants from the sun, photosynthesis, and a lot of kind of systems. Information flow coordinates body functions. Like, for example, DNA, it gives instructions to the cell how they need to grow. So this information is important because it will define how the organism will grow, how the organism will work, how the mechanisms and functions will work. Okay. Homeostasis maintains internal stability. Every organism needs the homeostasis. That what it means. Homeostasis, we'll talk later, but a slight introduction, is organisms that survive in challenging habitats need to keep their entire internal environment relatively stable. Stable is not equilibrium. We will talk about this later, but he needs to equilibrate all the system to keep alive and keep growing and keep doing things they need. Okay, so now we go to homeostasis, that is explanation about how it works. Okay, homeostasis is the stability of various physiological functions. And for example, body temperature, blood pressure, and heart rate. So homeostasis work to keep all this stable, all the systems stable. Let's check some topics about this. I'm sorry. Just, okay. So this is part of homeostasis, okay? And for example, what is the body internal environment that we are talking? In body internal environment is that, for example, extracell fluid or interstitial fluid, and intracellular fluid and plasma in the blood. So why this is an environment? Because you need to, they need to exchange material between the outside place of the cell and inside. So some things need to enter the cell, some things need to go out of the cell. So you need this environment, and you need homeostasis to make this kind of exchange stable. Okay. Sorry. Just a little thing about homeostasis. Here on this flow chart is the basic of homeostasis. For example, an organism in homeostasis suffers internal or external change. Okay. When this occurs, internal change results in loss of homeostasis. Why? Because if you increase the body temperature, this is not homeostasis. Yeah, the body is not in homeostasis because there is a disequilibrium. It's not stable. So the body needs to work to stable again, make it stable again. So the organism attempt to compensate this temperature. It means it will use a lot of energy to back to the stable back to stability. Okay. If the organism fails to do this homeostasis, to do this stable, there is an illness or disease detected. If the organism succeeds, so homeostasis is okay. So it's working, everything's fine. Okay. Okay, homeostasis depends on mass balance. It means that we talked about the exchange between extrafluid and extracell fluid and intrafluid of the cell. So this is external environment, cell, and external cellular fluid, internal cellular fluid. So they need to exchange mass, they need to exchange some materials, and all this about the balance. Okay, for the whole body, the total amount of substance in your body is your intake, like water, food, and something like this, plus your production, that the conversion of this food and water, excretion, and metabolism, minus expression minus metabolism. So this equation will keep your body stable. Okay. So there's two output options to maintain the mass balance that we talk now. Excretion is defined as elimination of material from the body, usually through the urine, feces, lungs, or skin. Okay, in skin by sweat, lungs, by volatile fluid and gas, and urine, feces, solid. Okay, and the second output is the maintaining mass balance is to convert the substance to a different substance through metabolism. For example, fat. if you the body to maintain the body stable needs to convert all the exceeds in fat, for example. So this will keep the balance, but any exceed is good. Okay, this is an example of intravenous infusion because it's the example how the body will absorb all the drugs you need to intake. So there's you cannot put 10 grams here. Just have an equation to know how the metabolism will proceed with these drugs. So you need to care about this. Okay, excretion clear substance from the body, and how to monitor substance from the body. In the primary clearance organs. The primary clearance organs is liver and kidney that deals with your blood, urine, and need to how can I say? Need to treat your blood to take out all the things that is not good for your organism. There is an indirect measurement, the clearance, that's the rate you can calculate. The volume of blood cleared of substance. So you take a sample of blood and how many times the substance needs to disappear from your blood. So it's per unit of time. So you can calculate it's a kind of measurement that you can do. You can do this with saliva, breast milk, sweat, and hair. These are elements that already have passed by this process. Okay. And this is important. Homeostasis does not mean equilibrium. Why this? It's stability, but not equilibrium, because if you look at some elements like calcium, sodium, and something like this, it's not in equilibrium in the extracell fluid and in the intracell fluid. There is difference between them. So you cannot say there is equilibrium. It's stable. It's working, but it's not in equilibrium because you need to analyze everything to say that it's equilibrium. Okay. So about the control systems and homeostasis. Control systems and homeostasis. Let's check some topics about this. Local and reflex control. So local control is about, for example, in tissue, it will happen only in a specific part of your body. Doesn't need to travel long distance in your body to solve a problem. For example, if a muscle or tissue needs more oxygen and suffering of because there is no oxygen, some chemicals will be released by the neighborhood cells and will dilate the vessels. And with this, the oxygen will flow in a higher concentration, and it will give more oxygen to the tissue or to the muscle or anything else. Okay, this is local. Doesn't need to use long distance. It's located in the local. As I say, local control is restricted to tissue. Near cell or group of cells senses the change in their intermediate vicinity. Response usually by releasing chemical, and response is restricted to the region. So, as I say, all the things will occur in the specific region. So, is that different from reflex control that uses long distance signal? And this kind of reflex control uses nerve system to travel, and there is an example here about an aquarium. So you have the water and thermometer and wire and the central and the warmer here, the heater. So if the water needs to be at 30 degrees, if past to 31 or 32, this is a stimulus. This stimulus will be detected by the thermometer and will send by wire that represents the nervous systems. and to the central that will process this and send an information in response to heat the water. Okay, so it will make more stable until you get stable this system. It means until you get back to 30 degrees in the water. Okay, is the response loops begin with the stimulus. So as I say, stimulus goes to the sensor. There is an input signal. So the integrating center will process this information and the output signal. So there is a target, there is the heater, and there is a response. Is that the water is getting warmer. Okay. So there is a flow chart about this, as I said. Negative feedbacks loops are not homeostatic. Why? Because negative feedback, for example, acceptable ranges is 30 degrees plus 1 or minus 1. So you have 30 degrees and the temperature grows to 1. So you have a negative feedback that needs a response to slow down this temperature. Okay. So it will generate a response to take an action in this place. Okay, there is another one here, another flowchart. Initial stimulus, response, and stimulus. It goes back here because for example, the water is not hot enough. So you back and is a loop until you get to the final temperature that you want. Okay. If you finally get the temperature, the feedback will go to the response and it will stops until need again. Okay. Positive feedback loops are not homeostatic. Why? Because, for example, in the birth, the the cervical stretch stimulates by oxytocin release, and it stimulates uterine contraction, and is just growing and reinforcing this action. It's not stopping. So this is a positive feedback. Positive feedback will enter the loop and doesn't stop until something different occurs, something that is needed for your body. So when the child birth, it's not going to the loop. It's go outside of the loop. Okay, the child just birthed, so you can send this message to your central of information, and this will stop. This will stop the oxytocin release, and this will stop the process. Okay, so that's why positive feedback are not homeostatic, because they're not trying to get stable. It's just to reinforce the action. Okay. The body will have biological rhythms result from change in set point. Everyone has some step points in your body. Like, for example, in the morning, your temperature is not as high as you need, or you're studying in the late night and your hands getting cold, because it's time to your body gets your temperature lower. So it's some body functions. For example, it's called circadian rhythms. Okay, and there is a graph between about, for example, midnight, noon, day and night. So the oscillation of the temperature of the body. So okay, so there is a circadian rhythm. And this is about cortisol. There is a difference too. In the day is higher and the night is lower. So this is rhythms as a signature of your body. Okay. So in the last topic is the science of physiology. We have some considerations here, final considerations. For example, good scientific experiments must be carefully designed. For example, in the case of a bird food intake experiment, there is independent variable and dependent variable. If you need to know if the lowest temperature will make the bird eat more, and you make an experiment, then you alter the temperature, and you can measure how much food the bird is eating. So the independent variable is temperature, because you can control. Independent variable is food intake, because that is what you are studying, what you want to know. You need a control group with natural parameters, and you need to make replication of this experiment, because to prove your results, you need to know a lot of experiments, and a model with substantial evidence may become a scientific theory. To become a scientific theory, you need to be replicated for a lot of times and giving the same results. Okay. The results of human experiments can be difficult to interpret because of variability, psychological factors, and ethical considerations. Variability is because a lot of humans have a lot of different conditions in the body. Everybody is different, and it can affect the results. And psychological factors like placebo and nocebo, that when you give a placebo to a person and say that it will solve some problem, probably the incidence of they will relate this medicine with the problem solved, and actually it's just a placebo, it's just sugar or something else. And nocebo is when you say to people that it will cause some side effects, and even if there is no drugs in the pills, people will report the side effects. Okay. And the ethical considerations is about testing humans, so testing with cancer patients, and the advantage or disadvantage to use humans in your experiments. Okay. Human studies can take many forms, a lot of results, and by many different groups studying. So just a small but potentially significant difference in the design experiment can cause problems. And that's why a lot of groups use meta-analysis, that's combined data that will use sophisticated statistics to analyze all the data generated by a lot of groups and get incorporation to analyze the results. Okay, so this is the presentation. Thank you so much.